The overall goal of this procedure is to measure epithelial barrier permeability in human intestinal organoids over time.
This method can help answer key questions in the gastroenterology field, such as how specific toxins, bacterial products, or even pharmacologic treatments can alter epithelial barrier permeability. The main advantage of this technique is that it allows for both visualization and quantitative measure of epithelial barrier permeability in real time and using human intestinal epithelial tissue.
Prior to microinjecting human intestinal organoids, the microinjector must be set up and the microcapillary prepared, installed, and tested as described in the text protocol. To fill the microcapillary with the injection material, submerge the tip of the glass microcapillary in the phytidextran injection suspension, being careful to avoid breaking the tip of the microcapillary against the side or bottom of the tube. Pull back on the mineral oil syringe to draw approximately 10 microliters of the phytidextran suspension into the microcapillary. When the microinjection suspension fills 90% of the length of the glass microcapillary, stop filling the microcapillary. Depress the syringe slightly before withdrawing the microcapillary from the microinjection solution to ensure that the tip of the microcapillary does not contain pockets of air. Remove the human intestinal organoid culture plate from the cell culture incubator and transfer to the biosafety cabinet with the microinjector. Remove the lid from the culture plate and center the first well on the microscope stage so that it is clearly visible through the eyepiece of the scope at the lowest magnification setting.